Hello, welcome to 13 Universal. This is A-level CIE Biology as we prepare for the May-June papers 2024. We are starting revision from immunities for paper 2. So this is series 1 and I am schedule. So get your writing materials. Let's solve some questions. Let's fry some fish. Okay. Cells of the immune system respond to the presence of non-self antigens. Okay, outline the features of non-self antigens. So pause this video and write out the features. Outline means you should give the main facts about non-self. Okay, pause the video. Welcome back. Non-self, you see non-self antigen. The features include, for the fact that it's non-self, that means they are not body antigens. They are antigens um, found on pathogens, okay, or infected body cells, okay. So usually they stimulate immune response, okay. But don't forget that antigens are um, macromolecules found on the surface of cells, but this is non-self, so it's found on the surface of pathogens or infected body cell. So if you look at this cell now, you can see we can find something like this on it, whatever. That structure there is antigen. This this is a pathogen. The entire thing is a pathogen, but the surface molecule is an antigen. So they are found on the surface of pathogens or infected body cells. But they stimulate immune response, so they lead to the production of antibodies. Okay, so antigens stimulate immune response, which causes the production of antibodies specific to the antigen, right? So the antigen, once it appears, then the lymphocytes that have complementary shape, whose receptor is complementary to the antigens, are usually stimulated. Okay, so the first thing to write is that the innate immune response, they are found on antigen, they are found on pathogens or infected body cells. They're usually proteins or glycoproteins. Okay, so they cause the formation of antibodies that are specific to them. So look at it. Yeah, stimulate an immune response, usually proteins. These are the features. They are recognized by, so they activate. This is how they cause immune response. Okay, so they activate or stimulate either phagocytes or macrophage neutrophils. Okay, they can as well activate B or T lymphocytes. Ignore plasma cells because B lymphocytes are first activated and then they differentiate to form plasma cells. Ideal leads to production of antigen presenting cells. Yes, so when they are found on the surface of macrophages, they are for macrophages are called antigen cells. So macrophages can actually present antigens of pathogens to stimulate lymphocytes. Okay, so they cause production of specific antibody. You can see that all of this. All right, so that's it. So you see all of this also. So they are found, all right, on they are not found on body cells, but they are found on pathogens, affected body cells, tumor cells, bacterial cells, wall, all of these, they make up antigens. Okay, let's go to the next question, guys. Yeah, four different types of cells of the immune system are shown in table 3.1. Complete table 3.1. Use a tick if the description applies to the named cells of the immune systems. Use a cross if the description does not apply. So you can see that. So pause the video and work on this. Okay, so take it. We look at this. So make sure you're working on this. Description of cells, able to go through number of cell cycles. Clonal expansion is by mitosis. The lymphocyte can divide, yes. So this is yes. Plasma cells do not divide, no. TFR cells can divide, yes. T killer, yes. So yes, no, yes, yes. Main role to secrete cytokines. The only ones that secrete cytokines among these four cells is T helper. So cytokines, when stimulated, when secreted, it stimulates B lymphocytes to divide to form phagocytes and to form plasma cells and memory cells. So plasma cells secrete antibodies, right? While they can also stimulate, cytokines also stimulate macrophages to carry out phagocytic phagocytosis, aggressive phagocytosis. So look at this. Presenting during primary immune response, okay? So they are present. All of them are present. So look at it. Yeah. So you can see that. Okay. So let's go to the next question, guys. Format. Human cytomegalovirus is a common virus affecting humans. In people with a fully functioning immune system, infection by HCMV usually causes no or only mild symptoms. Fig 2.1 is a diagram of a section through HCMV. In Fig 2.1b, only the outer part of the HCMV is sectioned. You can see that. Okay. So the same virus, okay, but different views, orientation. All right, just like mentioned here. This A is sectioned through this. V is the outer coat. The viral DNA section contains genes that code for proteins. So this contains genes that code for proteins important in viral replication and viral structure, including viral DNA polymerase. So even viruses have DNA polymerase. And proteins known as tegument proteins. So viruses can only replicate in host cells as they need to use processes and content of the host cell. Complete viral particles that are released from the host cell are known as virions. This is why viruses are said to be obligate intracellular parasites. They hijack the host cell machinery, metabolic machinery, to replicate their DNA and make their own proteins. Okay, so after a person has been infected with HMV, the virus remains in a dormant stage in the body for life. So they just lock in there, waiting for opportunity to take advantage. And when is that? So if the virus becomes active again or reactivated, the virus will only cause serious illness if the person has a weak immune system at that time. So they only show up when the immune system is weakened. So explain why the response to reactivation of HCMV is more likely to cause serious illness in a person who has a weak immune system. Okay, so I want you to pause this video and try this question. Why does it cause serious illness in a person who has a weak immune system? What does it mean to be weak, to have a weak immune system? Okay, so why does it cause serious illness? So pause the video and try it. Okay, when people have weak immune system, there are two things. Okay, you can say that they have fewer immune cells. So the immune cells, like the T helper cells, are few. So they release fewer cytokines, which stimulate fewer B lymphocytes, which make less antibodies. Okay, so they have fewer T killer cells, which burst fewer cells, pathogens, and infectable cells, or fewer phagocytes, which can have fewer. So these cells are fewer, right? And of course, because they are fewer, the immune response is slow. So there is low immune response. Secondary immune response is low, like primary immune response. Okay, these are the things that happen, why they cause serious infection. So because secondary immune response is low, so these viruses multiply faster and then destroy immune response. Okay, so look at it. See, okay, they start from this reference to time delay immune response. takes longer time for immune response to. So this is describing why weak immune response cause serious infection, because there's time delay immune response compared to normal. So if the immune system is strong, so especially for secondary immune response, there will be a rapid immune response. Okay, because we have more memory cells from there. So, but you see, because it's weak, there are few of these cells, so it takes longer time for immune system to respond. Slower secondary immune response. Immune response similar to primary. So, you see, secondary immune response is primary. Okay, primary takes longer time to occur because there are fewer cells. Now, secondary immune response is also taking longer time. It is taking longer time because there are fewer cells. Okay, look at it. As this is taking longer time, the virus is taking the advantage. So more time or easier for this HCM, HMCV to infect cells or replicate, spread to cause damage, any of these, as cells that reproduce virus carry out, okay, mitosis, so impact fewer immune systems, okay, in circulation, fewer macrophages, phagocytosis, fewer B lymphocytes, fewer memory cells, except no memory cells or longer present primary response. So because there are few immune memory cells, rapid response is not in view. So fewer B or T lymphocytes, T helper cells or T killer cells. Okay, look at this, guys. So less time, poor secondary immune response. Consequence, few immune response, release chances encounter. So now because there are few immune response, there are less chance to encounter recognition. So clonal selection or clonal recognition, clonal selection and clonal expansion very low, slow. Okay, that's why it's dangerous. 4 months. Now, cholera is a life-threatening and infectious disease caused by bacterium Vibrio cholerae. One of the symptoms of disease is severe diarrhea, severe rice water. Okay, people die 2 hours once they're infected. It's dangerous. Please run away from cholera, guys. All right, V cholera, these are different strains. 01 and V cholera 0139 are the two forms of pathogens that are associated with cholera epidemics. Large outbreaks. These two forms have different antigens that can be detected. So they have different surface macromolecule called antigen. However, rapid diagnostic testing RDT can be used to diagnose cholera by detecting the presence of V cholera. One type of RDT involves using a dipstick that contains mobile and immobilized monoclonal antibodies. Monoclonal antibodies MAB maps are specific in their action. Okay, yeah, you can see that. Please, I want to study this. This is test this direction. So this is inserted into the blood area where dipstick is inserted into the mixture of blood. So this area here, number one area contains two different mobile monoclonal antibodies, right, that can recognize O1. So anti-O1 antibody, anti-O. All right, remember that the antigens O1 and O3, they are in these Vibrio cholera pathogens. Okay, they are coming from the blood, but we already have, all right, antibodies that can recognize them. So they are here. So the one that can recognize O1 is called anti-O1 antibody. So they are monoclonals. Also, we have so we already made an antibody specific to these antigens. They are here, but they're not. They're mobile. Okay, so that means that if we insert this into blood, right? So let's assume you're inserting into this, test tube that has blood, yeah. If you insert it here, so once they come in contact with the blood, these antigens, all right, they start moving up, So when they come here, all right, they bind to the antibodies specific to them, because these antibodies are mobile, so they continue to move up. So they form, once they bind to these antibodies, they form antigen antibody complex for this and antigen antibody complex for this. Okay, when they get here, because they're moving up, now this is test area containing immobilized monoclonal antibodies that binds to antigen antibody complex. So we have another immobilized monoclonal antibody that recognizes antigen antibody complex 0139. So if antigen antibody complex is formed here, this will recognize them in number 2. That means that if this virus with this 0139 is present, all right, it's going to bind to the immobilized monoclonal antibodies, because if it is present, it will first of all bind to its monoclonal. Remember that when antigen binds to the antibody, they form antigen antibody complex. So that antigen antibody complex is now used to produce a monoclonal. There's a monoclonal that can identify this antigen antibody complex from here, and that is when you see a line here, know that this antigen antibody complex is being bound to by an immobilized monoclonal. Okay, but this one is specific to 0.139. Okay, so if this person has this virus here, all right, of course it's going to form antigen antibody, all right, but they're going to come here and they stay here. Some of them will stay here. Okay, they will stay here. Why? Because the monoclonal here binds to antigen antibody, all right, yeah, for this 0139. Okay, but if it's not there, if 0.0139 strain is not there, that means there will not be any line from here. Okay, but look at area 3. Test area contain immobilized. So you see that now, see, when this antigen binds to its antibody monoclonal, right, of course they will move up. There's another immobilized monoclonal antibody, but that monoclonal antibody is specific to antigen antibody, and that antigen antibody is an antigen antibody that has 01 as its antigen. Sorry. Okay, here. So if that strain is present, the line formed here means 01 is present. Control here means that, okay, because there are some of this antibody, right, let's assume there's no antigen, right, present here. Okay, so what happens is that the mobile monoclonal antibodies will still move up. When they come here, we have monoclonal antibodies that can bind to the mobile, all right, monoclonal antibody, sorry. So they will still form line here. This shows that this strip is working, because let's assume there's no virus present here. What happens is that this mobile, right, against this antigen will move up. When they get to this area, we already have immobilized antibodies for them that can bind to them, so they form a line here. So there will always be 2 lines. One here, this line, any line from here tells that the test strip is working. Okay, if you have another line here, it means that what is present is strain 01. Okay, now if another line here, then 013 strain is present. So if I have 3 lines, 1, 2, 3, that means that the 2 viruses are present. But if none of the virus is present, we'll always have control area, because it shows that the test strip is working. But if there's no line here, that means test strip is not working. Okay, so if you have a line here, this is positive for 01. Have a line here positive for 0139. Okay, so now with this in mind, come on, let's go do this. Destroy some questions. So fig 2.1 shows a simplified diagram of an RDT dipstick that can be used to distinguish between V. cholera 01 and V. cholera 0139. A fecal sample. Remember that cholera pathogens, they are transmitted through oral fecal route. Okay, when you eat food or drink water that is contaminated with the feces of an infected person. So that's what we are taking a fecal sample from a person which with suspected cholera is collected and added to a reagent solution to form the test mixture. Okay, so let's see next. All right, the test mixture moves up the dipstick like I showed you through area 1. All right, now when it gets to area 1, the mobile monoclonal antibodies are attached to tiny gold. See, that mobile ones has tiny gold, okay, particles. If these antibodies collect in test area 2 or 3, a colored band become visible, okay, meaning they are present, the strains 01, 0139. A colored band that becomes visible in area 4 confirms that the test strip is working and that the results are valid. explain how the structure of monoclonal antibodies in the dipstick allows them to be specific in the action. Right? So pause this video, try to solve this question. Remember that we are here working to solve questions. Okay? So why is it that explain, give reason how the structure of the monoclonal antibodies in the dipstick allow them to be specific in the action. Okay, now remember guys that if you remember the structure of an antibody, antibody has it has four, okay, this is 4 polynucleotide. Okay, the short ones and the long ones. Okay, so 2 short ones and 2 long ones. This is joined by disulfide. This is joined. This is disulfide I'm drawing. So 2 long ones are heavy chains joined by disulfide. So guys, so look at this. This part is called the variable region. Of course, this space is antigen binding site. This is also the variable region. This space is antigen binding site. So now antibodies are just different from others based on the antigen binding site. Okay, this antigen binding site has unique primary structure of amino or amino acid sequence that gives it a unique shape, right, that allows specific antigen to bind. Okay, of course, here is the constant region that binds the receptor on phagocytes. This place is called, you see this place? This is called hinge region that allows the antibody to bind to antigen. It gives the antibodies flexibility. Hinge region gives flexibility. Now this is bond, disulfide bridge that joins 2 long chains or heavy chains. This is the disulfide bond that joins 2 short chains. A short and a long. Also disulfide bridge. Okay. So the question is explain how the structure of the monoclonal antibodies allows them, okay, to be specific, because the variable region of the monoclonal antibody is specific to a particular antigen. So it is complementary to a particular antigen. Why? Because of the 3D shape, which is as a result of the primary structure. The primary structure, that's amino acid sequence, affect the 3D shape, okay, which now gives it a particular, allows it to bind to specific antigen. So now that therefore antibodies that are specific to O1 antigen binds to it, okay, and they are only specific to O1 antigen of this virus. The ones specific to O139, okay, binds only O139. Yeah, so look at it here. Allow antibody, okay, it's a reference to ignore reference receptor, ignore active site. So you don't call antigen binding site of an antibody. You do not call it receptor. Guys, don't call this. This is antigen binding site or variable region. Don't call it receptor. Receptor, all right, is for cell signal. Okay, active site is for enzyme. Ignore, and you see, I means ignore. Okay, don't say combines or bonds. Okay, now see what you say. Specific. So this antibody, okay, is specific or different. Okay, or can say specific different pathogens antigen binding site. So that's specific. So binding site for antigen. So reject antibodies described as enzyme. Antibodies, they don't describe as enzyme. They don't have anti-active site. So accept each monoclonal binds to specific or particular pathogen antigen. Yeah. So each monoclonal has specific or different tertiary or variable region, that is this. Okay, due to primary structure, sequence of amino acid, binding site and antigen have complementary shape. So you call it binding site, anti-binding site. Okay, that is why they can bind to specific antigen, guys. 2 marks. All right. Fig 2.2 shows the results of samples taken from 2 different people, A and B, who are suspected of having cholera. Okay, can you see? Now, with reference, if you're a doctor and you run a sample, and then this shows out, what does it tell you? What does it tell you? With the knowledge of what we have done so far, with reference to Fig 2.2, state and explain the conclusion that can be drawn from the result of the RT dipstick for each person. Now this is person A, person B. You are the doctor sitting across, all right, these guys are sitting across your desk, all right. You run a test for person A, person B, keep away sentiment, whether they are related to you or not. What does this tell you? Okay, now of course you can see, pause this video and answer this question. Okay, pause it and try. Now look at this. This area shows control. From the beginning we said that means that this test is working well because they have immobilized monoclonal antibodies for those, mobile monoclonal. So we have immobilized monoclonal against mobile monoclonal. So when they bind to the form, it's showing that is moving upward. Okay. Now this 3 is for O1. Remember. So this shows that this person A is positive for having the pathogen with O1 antigen. Okay. So it doesn't have 0139. This is for 0139. Okay. All right. So yeah, this person is negative. Is negative. It doesn't have any of those pathogens because there's no line here. No line here. If there's one here, that's 0139. There's one 01. Okay. So based on what we have said, do you get? So you can see that person A has cholera, is infected with viral cholera. So because this is where we immobilize antibody, all right, against antigen antibody complex of O1. This 3 region from the beginning. So if it forms a line here, that means that person A has cholera O1, is infected with cholera O1, and person B does not have. Okay, does not have. All right. As a reference to person testing positive for cholera, and then negative. Tests are valid, working because control. So test. So because we have control 4 here, they're valid. Person A banned color. Of course, we're explaining it now. 3 because anti-O1 mobile antibody. This anti-O1 mobile antibody binds to specific antigen on Vibro O1. Okay, and then accept antigen antibody complex O1 formed. They were formed here, but they're still moving up. Okay, so what happens? Okay, so they were formed here. So when they now got here, that antigen antibody complex now forms another complex with immobilized antibody. All right, so you see person B, no band, no color, not because no antibody binds with antigen. Okay, no antibody comes. Good. 3 marks, 3 solid marks. Let's roll it. 4. Plasma cells can be used in the commercial production of some monoclonal antibodies. The method of production is known as the hybridoma method. Outline the steps in the production of monoclonal antibodies by the hybridoma method. So outline the steps. This question is always coming out, guys. So pause this video and try. Pause the video and try the questions. Okay. Right. Very importantly, guys. Remember, antibodies are only produced against specific antigen. So if you want to produce monoclonal antibodies, the first thing that you do is that the antigen that you desire to make the monoclonal antibody for. So the antigen is injected into a small mammal, like a rabbit or mouse. Okay, so inject the antigen into this mouse, rabbit or mammal, and then allow the next immune response is allowed to occur within several weeks, like 2 weeks. Okay, so after immune response has occurred, the next thing that happens is that the plasma cells formed in this mammal is extracted from the spleen, right? So and then the next thing that happens is that this plasma cell is fused with a cancerous cell like myeloma. All right, so we're using a chemical called fusogen to form hybridoma. So the hybridoma is screened. How do you screen it? Put it inside the well and introduce the antigen. So if that is the right hybridoma, when you introduce the antigen, the hybridoma will make antibody which forms agglutination, right? Binds to the antigen and forms antigen antibody complex. Okay, all right. Now, of course, the hybridoma can now be grown commercially in large meters. As it grows, it makes large quantities of antibody. Remember that plasma cells they cannot divide, but they make antibodies, and cancerous cells divide indefinitely. So that's the first thing you do. So inject, small mammals injected with antigen. The next one, immune response occurs. You allow immune response to occur. All right, plasma cells of the lymphocyte, or called the splenocyte, extracted from spleen, and of course plasma cells, all right, activated being lymphocytes and then infused with myeloma or tumor or cancer cells. Why? To form. Why do you fuse it? Plasma cells make antibodies, but they cannot divide. Okay, myeloma does not make antibody or tumor cells, but they can divide indefinitely, and we want large quantities. So if you fuse two of them, so hybridoma has both features. They can make antibodies and they can divide indefinitely to make. They continue to make large quantities of monoclonals. They continue to make it. All right, for us. Okay, hybridoma cells separated into wells to produce clones. So you clone them and then screen them. Okay, hybridoma desired monoclonal antibodies, screen them by adding, okay, the antibody, then large scale production, and then we're able to save lives. Yeah, antibodies can be collected from human blood donors and used to treat people that may have been infected with a pathogen. This prevents them from becoming ill with disease. So they say that if you treat people with sickness, treat them with antibody, they can be saved. Why? Explain why this treatment does not prevent people from becoming ill if they're infected again with the same pathogen. So if you treat somebody with an antibody against a pathogen, just injecting it like anti-rabies, somebody's bitten by mad dogs. If you've been bitten by a dog that has rabies, okay, or somebody's bitten by a venomous snake like black mamba, so your immune system cannot save you on that day. We need to inject you with antivenom. This antivenom is just antibodies made from another organism, okay, to save your life. Now, why is it that this treatment does not prevent people from becoming ill? So if the person is bitten by black mamba again, will the person survive? No, that's the answer. Why? Pause the video and write it. Connect this to the template content of this topic. So they're just asking you, why is it that immunity provided by antibodies made by another organism does not give temporal and permanent, long lasting? Okay, that's what they're asking you. They're asking you to talk about passive immunity. Okay, that's what this question is asking you. So passive immunity, the antibody is not made by the person that is ill. So no memory cells are formed. So the antibodies are not made by the immune system of the patient, okay? Because no immune response occurred. So no memory cells formed, okay? So that means that in the future, when the same pathogen enters the body, there are no memory cells formed. So it's going to take long time for immune response to respond. So it's going to go through the primary immune response. When we have primary immune response, okay, it takes longer time because there are fewer memory cells that can recognize the antigen on the pathogen. Okay, so remember that antibodies you're injected in this person can be digested by the body. Look at it. So this is artificial passive immunity, not natural. So no immune response. This is why it cannot protect the person. Okay. Idea that antibodies are broken down. Yeah, they're destroyed by the body. No long-term, just short-term temporal. Memory cells are not produced. Memory cells only produce in active immunity during immune response. Okay, after production of antigen. So no clonal selection, no expansion, no antibodies produced by the body. So if a baby has been protected by the mother's antibodies in the breast milk, that baby needs to be vaccinated so that we can form active immunity. Is that okay? This is why children have to be vaccinated so that they can develop their own active immunity. But if they are injected, if they have the mother's antibodies in their body, the mother's antibody will soon be digested or degraded by the enzymes in the children's body, and the children are not yet protected. If you have a child, or you have a younger one, okay, tell your mom to vaccinate them. Okay, all right. Yeah, blood plasma plays an important role in the transport of molecules to antibodies. Scientists discovered that some of the antibodies in the blood plasma of sharks have a different structure, okay, to the antibodies found in human blood plasma. Fig 2.2 shows the structure of an antibody molecule found in the blood plasma of a shark. Yeah, shark. Shark. You know that song they sing, baby shark, dun, 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 mommy shark, daddy shark, dun. Okay, that helps you to remember this. So this is the structure of antibody from shark. Who knows that shark will be used by human beings? Antibodies from shark to save life. Question A, state how quaternary structure of a human antibody molecule differs from quaternary structure of the shark. So quaternary structure of human antibody, how does it differ from quaternary structure of the molecule or shark antibody? This is shark antibody. How does it differ? Pause this video and try. Okay, pause it. Now this is the quaternary structure of human antibody. Okay, we've drawn before. All right, just simple difference between them. Simple difference. Tell me that difference, my friend. You can see, yeah. So you can see that this one here, this is human antibody, this is shark antibody. Okay. The difference is that look at the quaternary structure. Quaternary structure is the presence of more than one polypeptide chain. So this has only two polypeptide chain, this and that. Okay. Why these four polypeptide chains? One, two long chains and two short chains. Okay, two long chains and two short. Okay, so that is it, guys. Yeah, human antibodies are made of four polypeptide chains, two light or heavy and two short. Yeah, two light is the short ones. Okay, long is the heavy ones. All right, one mark. Yeah, human antibodies are used in the treatment of some forms of cancer. However, read this question, guys. So, however, the antibodies injected into the bloodstream can only reach small percentage of the cancer cells that form in cancerous tumor. Shark antibodies are smaller than human antibodies. Scientists are researching the possibility of injecting shark antibodies into the bloodstream to treat cancerous tumors in humans. Sharks being used to save humans. That's amazing. So just how using the smaller shark antibodies may be more effective in reaching a greater percentage of cancer cells than human antibodies, okay, and lead to greater success of treating cancer. All right, pause the video and try to answer this question, my people. Okay, so you see, remember, this is capillary, okay, this is, see, because we're injecting this into the blood, so but the blood, right, carries stuff to the tissues. So blood transport these antibodies to the tissues. So this is blood capillary, okay? Right? So red blood cells are here, squeezing through, okay? Remember. So we now have body cells, the body tissues here, okay? Body tissues, you can see them. Body tissues, you can see body tissues, okay? Yeah. So now, so what's the advantage of using shark's antibody? So one of the advantages of using shark antibodies is that if for example, we have, this is where the tumors are, okay, and the shark antibodies inside the capillary, this capillary, okay, red blood cells are here. So shark antibodies are small, so they can easily live through the endothelial pores of capillary wall into the intertissue space where the tumors are, okay. So more of them can live the blood through the endothelial pores, and then they can bind to the tumor cells, okay, and then destroy the tumors. So it's more effective because they're able to pass through the pores, so they're able to bind more to tumors because many of them are present, and they're able to, you know what antibodies do, tag tumors, so that they can be engulfed by phagocytes. Okay, so if you check the masking here, so more easier to pass through the gaps in the capillary wall. These are the gaps between the capillary walls and the endothelium. So more easy to enter, okay, surrounding cells. So they're able to enter the surrounding cells. Okay, can cell destroy quickly. Cancer cells destroy quickly because they're small. So more of them will enter. So may not, and that is they may not trigger immune response because they're small. Easier to diffuse, move through the tumor to reach all cells. Binds more tightly, so they bind more tightly to antigens on cancer cells, and then macrophages respond stronger than overall size. Okay, when drugs are attached, so they can easily deliver drugs to all the tumors here and tumors there. Okay, because antibodies are used for treatment, monoclonal antibodies. Okay, they deliver drugs and then allow selective toxicity. Okay, too much. Antibodies can also be used in the prevention of infectious disease. Explain how injection of antibodies into the bloodstream can protect a person from disease after infection by a pathogen. So if somebody is infected by a pathogen, all right, somebody, that's what's called anti-tetanus. You know, you are wounded by tetanus, by injury, and tetanus is your blood. How do we protect you? Okay, pause this video and guys, retrieve the information on the mode of action of antibodies during immune response. Okay, how does antibodies, or how do antibodies protect a person from a disease? How? Okay, number one, antibodies will bind to the antigens on these pathogens, okay, using their variable region. Their variable regions specific to the antigen, so they bind to the antigen. Now remember that they are produced by immune response. Due to immune response, they are released. Okay, but in this case, we're injecting this person with antibodies, so they are not even, immune response is not occurring. But now these people are injected with antibodies. The antibodies bind to the antigens, right, because they're complementary to the antigens on the pathogens, and then they can burst the antigen or coat the antigens, mark them out for phagocytosis, call it opsonization. They can as well clump up the pathogens, prevent them from spreading. Okay, that's called agglutination, right? Now, if the pathogens release toxins, they can neutralize toxins by making them insoluble. So these are these. So that's called artificial passive immunity. That's the name. So you know, they're very fast. Okay, immediate, fast, quick response time. Antibody binds to pathogen, and when it binds to it, it starts the work. Binds to toxin, neutralizing them. Yes, all antibodies act as antitoxin. That's it. So reference phagocytosis, opsonization, binds to flagella and immobilize pathogens. Okay, clump them up. Antibodies cause agglutination, clumping them. Okay, pretty much. So thank you for watching through this particular series. We are going to series 2. See you, and I wish you the best in your May June exams. Okay, watch all our videos. We are bringing out more topicals. Okay, bye. This is Shedro from 13 Universal.